really interesting, and it's a lot of fun. So anyways, I wanted to now go into endogenous retroviruses because these points have been brought up lately on viruses in general, guys. And I put out a video today that I'll play for anybody who hasn't seen it. But there's been some critics, one specific critic on my SFT Facebook page, who I said, instead of the comment war, let's have a live debate. He doesn't want to do it. The name he goes by, at least on Facebook, is Barry. And he's written blogs and blogs and blogs on ERVs. And I'm happy to debate him on it live, okay? And he was having a back and forth with one of the brothers, Redefine Living. And he was saying, how do you explain that the Phoenix experiment, there's an experiment, okay, that he's looking to, supposedly resurrected fully replication competent retroviruses. Okay, his blog, that specific article is titled "The Phoenix Virus: An Explanation of an Experiment." But it's so funny, and this is why I want to get this guy in a live debate. Okay, this is why I want to get him in a live debate, because this is where you can call them out for tap dancing. This is where you can call them out for dodging and also misrepresenting. I've pointed out in my numerous debates with popular YouTubers too, like Conspiracy Cats, Team Skeptic. Okay, I've debated biologists on this. One biologist I debated on T-Jump's channel. We spent 30 minutes on ERVs. And I said, the question is, okay, guys, and remember this. And also let me know how my audio is too, because I'm trying something new. So the question is this: are these endogenous retroviruses, okay, these various classes of retrotransposons, are they really the ancient remnants of viral infections, okay, that have been passed down? Or are they created units of DNA function? If they really are created units of DNA function, what would we predict? Well, function, of course. Okay, so this critic, Barry, okay, his blog is named barryhisblog.blogspot.com, okay, all easily debunked. Actually, he bought my book. Thanks for the support. He left a review and mainly just focused on endogenous retroviruses and ignored everything else confirming independent origins, which is fine. But he completely misunderstands this point and this question that I just went over right now, okay? Because the question is not whether endogenous retroviral-like sequences can actually form viruses, because I've discussed, and I've even discussed in my books, that there is a creationist hypothesis. I look to these elements as variation-inducing genetic elements. They're beneficial. They were created to be helpful and beneficial and functional. Again, I recommend my debate with Conspiracy Cats, where the whole debate was pretty much ERVs almost. Okay? So the origin, according to at least one of our hypotheses, the origin of these viruses, okay, post-fall, is that they arose from endogenous retroviral-like sequences in our genome, instead of viruses coming into our genome from the outside. Are they coming from the inside? Are they created? Or are they coming from the outside? And this was this Barry guy's best argument. And I say, come debate me live. Okay, he wants to take me away from that which is important into an endless Facebook comment war. Okay? So that's the question, from inside or from outside? The evidence seems to suggest that our genome is the source of viruses to begin with. So here, I'm going to ask it again. This is what the question should be. So this Phoenix experiment doesn't help differentiate between the models. The question is whether our ERV-like sequences are functional. And that Phoenix virus experiment doesn't even begin to address that question. Okay? Here's the thing, and here's what's important. And one debate that comes to mind was with Mark Drysdale, where he was all hung up on viruses. Why would God make viruses, especially viruses that are bad? And that's how we started off the discussion. And I pointed out the fact that the majority, guys, okay, you need to put away what you thought was true and use your critical thinking skills. Most viruses are actually beneficial and helpful to our bodies. And people want to say, but don't most viruses cause disease and illness? And I always point out the fact that no, viruses do not mostly cause disease. Okay? Here's the fact. There are actually more viruses, okay, than there are bacteria. For example, if you were swimming in the ocean, what that actually means is you are swimming with an ocean full of bacteria. And what's funny is there are more viruses than bacteria. So what are the viruses doing? Well, they are controlling and regulating the amount of bacteria. It's ecosystem balance. This is important. You would not want to live on a world without viruses or without bacteria. Now, you've heard, okay, that we also have more bacteria in and on our bodies than we do with cells. And we have trillions and trillions of cells. And yet we have even more viruses than bacteria. So why are we not all dying within an hour if viruses are bad? No, they're beneficial, they're helpful, they were created for purpose, for benefits. They're doing the same thing that they're doing in the ecosystem of, say, the ocean. They're controlling, they are regulating. It's all about control and regulation of the number of bacteria in our gut. I take probiotics. A lot of people take probiotics. Good bacteria, that's good for you. Especially when you just got done a round of antibiotics where it's killing all your good and all your bad bacteria. You should take then a round of probiotics. See how the chat's doing here. And okay, good questions, good comments. One question from Luca. And JJ says, I always said ERVs are rogue information. Yeah, so Luca, once again, that's not the point. Is we know how to recognize viruses. We know how to recognize viral-like elements. Just like this apparent Phoenix experiment, just like the lines of evidence that the evolutionists want to put forth as evidence for endogenous retroviruses, for example, being the result of universal common ancestry, completely misses the point. Are they the result of ancient viral infections, or are they created units of DNA function? Okay, those are the important questions. We would predict function and purpose and benefit, okay, to these DNA elements. Now, recognizing them, that's not differentiating, because that's just recognizing what we already agree is there. The question is, did they come from the inside or did they come from the outside? But you might have asked that question actually before I talked about that, so that's fine. Good question. Okay, so remember, we have more viruses in our bodies than we do bacteria and cells. It's about regulation. The viruses are regulating the massive, massive numbers of bacteria in our bodies, in the ecosystems. Guys, without bacteria, for example, let's focus on bacteria. Without bacteria in the oceans, well, without bacteria in our bodies, we wouldn't exist. You wouldn't want to live in a world without bacteria and viruses. When it comes to the ocean, you wouldn't even want to swim in the water because there would be such incredible imbalance in that ecosystem. Viruses are highly beneficial. Bacteria are highly beneficial. Now, here's the thing. Not all viruses are good, but most of them are. Okay? Now, they can burn hot and fast due to mutation. They can cross species, like with what we're seeing today, with the big pandemic with the virus. Okay? When a virus jumps from its originally created space, okay, or arena, to a brand new environment or field that it can't recognize, that it's not used to, let's say into us as humans, then the immune system doesn't know how to deal with it. It's just jumping from one species to the next. And now that species, whatever it is, it doesn't exactly have the ability to regulate the virus properly. And that's why the virus then tends to burn hot and fast. The best thing that can happen to these viruses is that they mutate to oblivion. Okay? These viruses that look like ancient viral infections, we know what they look like. We know how to detect them. But they're actually very tightly regulated. Okay, guys? And to Luca, they're very tightly regulated into the genomes of living organisms, and they're highly, highly functional. The evidence is clear that they were created this way on purpose. Okay? So not all viruses cause disease. Okay? And I point out the fact that there are certain classes of retrotransposons. Ones that comes to mind is there's one certain class in the mouse embryo that when you deactivate it, okay, the mouse embryo, it's developing, you deactivate, and then it stops because it's dependent on the function of these retrotransposons. Okay, these are not junk. These are not worthless. These are not, based on those two questions, were they created? Are they created units of DNA function? That's what the evidence seems to suggest. And we can have internal and external escapees. The internal escapee can simply be due to a mutation. This is all evidence of genetic entropy. So the next time you hear the word virus, just think of something good, beneficial, and helpful. Okay? Now, when you hear the word disease, yes, think of something bad. But most viruses, most bacteria, they're not causing disease. They're not causing harm. Brother Montaz. Yeah, Lena Powell, great point. That's what's going to happen. It's going to mutate into oblivion. I found it so funny because Doki Doki, that's my mood too. I love it. Jungle says, where did it go? He made a good point. ERVs are rogue elements. Yep. Yeah, good point. Montaz, good point as well, brother. There's no way to refute the fact that viruses are helpful. There's no way to refute the fact that bacteria are helpful. They were created for good, okay? And that's what the evidence seemed to suggest, okay? Because the case is being made, okay, guys? Briefly, I'll touch on it. Now, there's a number of papers on variation-inducing genetic elements from Creationist, and I'd highly recommend looking into them, okay? But I'll briefly say that the viruses we see today, they've originated within us. They were created in us, okay? By cellular parts. Cells are packaging things in a variety of ways, okay? And then what happens? Oops. They've accidentally packaged something that is exactly what a virus is. So it's quite possible and logical that viruses came from our genetics, as compared to what most people think of, okay? We have what look like viruses in our genome. And as I pointed out, they're tightly regulated, highly functional. The ones that went bad and rogue, the internal escapees, they're the result of genetic entropy, the result of the fall. Endogenous retroviruses, there's no evidence for co-option. No evidence. I was debating a biologist on T-Jump's channel. I said, show me an example of a non-functional endogenous retrovirus going from non-functional to functional. I've asked all the team dodgeball, never been given an answer, nothing but dodging, because there is no answer. There's no empirical evidence. It's all forced, retrofitted. It's all inferred, just like the orphan genes, okay? No, this co-option is a fairy tale story invented to explain the data. These ERVs, they can act as DNA regulatory elements, long non-coding RNAs. They can act as triggers for the innate immune system. Okay? They're involved in immune responses. Function after function after function. They help accomplish many crucial functions such as gene expression, help in development, and a lot of the activity associated with these transposable elements, they're actually very tightly controlled by epigenetic mechanisms and specific RNA molecules. So it is ridiculous to claim, especially without evidence, nothing but imagination and storytelling. It is ridiculous and unscientific to say that this incredibly complex genetic system came about through ancient invasions of RNA viruses. No. Claiming they were co-opted, okay, it's nothing more than a story to retrofit the data. These ERVs, for example, let's focus just on ERVs, these crucial and critical functions involved in, say, regulating genes. I've got a paper here that says they're even involved in determining cell types. I talk about this with conspiracy cats. I actually have a clip I'll show you, because they'll often say, when I debated Mark Drysdale, no, this isn't true. This is correct, because they're just blindly believe what they were taught to believe. Okay? So when you tell them about how functional these retroviruses are, they dismiss it. And I'm going to show a clip, actually, where I've got a number of papers. Doki Doki, thank you so much. I appreciate the super chat. Good to see everybody. Wow, time flies by when you're having fun, guys. We've been going over an hour. So lots of good comments, good people. Thank you for the super stickers. So I hope this is helpful on endogenous retroviruses. I really wanted to hammer this one. I'm going to show some papers, actually, in 1 second here. Where can I go to find the papers? No, I'll screen share, and then we're also going to show a brief clip. And let's go right here, screen share. Let me know it can be seen. Okay, so let's see. Ton of slides here. So let's go to, okay, so let's get some papers here. Look at this one. Published August 19th, 2019. This has to do with the retrotransposons. Far from being junk DNA, the pervasive retrotransposons that populate the genome have a powerful capacity to influence genes and chromatin. A new study demonstrates how the transcription of one such element, HERVH, can modify the higher order 3D structure of chromatin during early primate development. It's so funny. Where these primates, these mammals, like with the mice, the embryo, then how they stop developing. Apparently they just weren't able to have babies. They weren't able to reproduce successfully and pass on their genes until they got this co-opted retrotransposon or endogenous retrovirus. So you'll find excuses and excuses with this Barry guy in his blogs. All the arguments are retrofitting or inferring or assuming ape to man common ancestry, assuming they were co-opted, not actually answering the question as to whether they really are the ancient remnants of viral infections, in that we've inherited these from a common ancestor, a primate common ancestor, a common ancestor with the chimpanzee, versus are they created units of DNA function? I've already spent 20 minutes, 30 minutes showing how important viruses are. And the same thing goes for viral-like elements too. So no, they can't explain this. It's exactly what, look at all these papers. Retroviral promoters in the human genome. These data suggest that ERVs may regulate human transcription on a large scale. Guys, when God created animal life, the animal types, Adam and Eve, he front-loaded them with a bunch of DNA differences that can lead to variation and adaptation, epigenetic mechanisms, and variation-inducing genetic elements, these viral-like elements, bacteria, okay, other functional DNA elements like ALUs, some of these pseudogenes that were thought to be genetic mistakes inherited from common ancestors. No, God has front-loaded the genomes of living organisms with so many beneficial and helpful DNA elements for adaptive purposes, epigenetic purposes. We have so many millions of genetic switches just waiting to be turned on or off via the environment. Function after function after function. Our genome's multidimensional. It's nested. Okay? You have messages that can be read both forwards and backwards. Okay? The DNA cannot be explained through evolutionary processes. And the forward-thinking mechanisms in the genome that we know exist, they can only be explained by a forward thinker unless the evolutionist wants to claim that natural selection and mutations and other evolutionary mechanisms have a mind. So let's see, more papers here. Taken together, our findings suggest that HERVs behave like normal cellular genes and are a permanent component of the transcriptome of a cell. Comprehensive analysis of human endogenous retrovirus transcriptional activity in human tissues with a retrovirus-specific microarray. Considering this, right here, endogenous non-retroviral RNA virus elements evidence a novel type of antiviral immunity. Function after function. Regulatory activities of transposable elements from conflicts to benefits. ALUs influence alternative splicing. Look at this. Scientists identified, it's not just one or two papers, one or two functions. When I was debating Erica for the second debate, it's just one or two. So therefore we can assume that they were co-opted because it's just one or two. And I'm like, it's not one or two. We're looking at genome-wide activity, highly, highly beneficial and functional, these elements. Look at this. Scientists identify new beneficial function of endogenous retroviruses in immune response. Isn't that funny? Endogenous retroviruses play a critical role in the body's immune. Just co-opted, this. No. Listen, contrary to what the evolutionist says, if it was really the origin of these was ancient viral infections and invasions, okay, well, it would likely disrupt the genome, disrupt the function, as compared to create or lead to a novel function, especially something as critical to the genome as development, cell stress responses, regulation, expression, okay, so on and so forth. So, endogenous retroviruses play a critical role in the body's immune defense against common bacterial and viral pathogens. Isn't that funny? Researchers have found. See how the chat's doing. Ian Chen. Ian, good to see you. We answered some of your arguments earlier. You'll have to go check that out. So Ian will not be able to show us any evidence for the fact that viruses have come from the outside. No. For 30 minutes, Ian just got here, so he didn't see it. We went over the huge amount of irrefutable evidence that viruses, bacteria, they have originated from within. Okay, they are used in the ocean. The viruses and bacteria, they're there for the ecosystem, the benefits of it. You do not want to live in a world without viruses and bacteria, put it that way. Thank you for posting that debate, Doki Doki. And Ian Chen. Ian, so we went over some predictions. Let's see if you can reiterate the history of civilization prediction that I've talked about in great detail. I even called you out on live in our video to Vice Rhino. Let's see if you can reiterate it, because you keep saying there's no predictions. Let's see if you can reiterate the predictions on the thousands of species, predictions in regards to genetic stamps in the history of civilization. So anyways, Yeah, so let's see evidence. Let's see evidence that these have all originated from the outside. You ain't going to find it. Ain't going to find it. Endogenous retroviruses function as gene expression regulatory elements during mammalian pre-implantation embryo development. I'm going to show you guys a good article here. Okay. Right here. No, not that one. Right here. So, variation-inducing genetic elements. Actually, before I read this one, guys, I'm going to play the quick clip. Just 1 second. All right, so I'm going to make sure the audio is on. Speed in the house. Him Snake, good to see you. Ian Chen, you are famous. You are famous for not being able to reiterate my points that I've made 1,000 times. You're famous. Own it. Rock it, my man. So, and here's what's funny. So Ian Chen, he's totally wrong, but he says, well, Jensen's made no predictions other than the prediction on the Khoisan people, which we don't know yet. Yeah, that's called a future testable prediction, Ian, where you make a prediction that only future observations and experiments can tell us whether it's true or false. That's called a prediction, okay, as compared to a retrodiction. Sometimes it takes years before predictions are actually fulfilled. But here's the thing. Dr. Jensen came out with a huge series and some technical papers confirming predictions on the Y chromosome. Gutsa Gibbon attempted and failed miserably to refute those predictions. I put out video after video showing why it's a misrepresentation, and Joshua Swamidas was probably unintentionally misrepresenting Jensen, or maybe intentionally, I don't know. So here's the thing. Here's the thing. There's been predictions that only future observations can tell us whether or not they're true. And there's been predictions that have actually already been shown to be true. And the evolutionists just aren't up to date on it. So it's just sad. But that's why, Ian, I'm looking forward to getting you into a live debate. Very excited. Very excited. Ian might want to come in right now, actually. You know what? Typically, I don't do that because that might... Ian, come in here and I want you to reiterate my explanation for Dr. Jensen's predictions on the history of civilization, not only in the Y chromosome, but also in the mitochondrial DNA when it comes to phylogenetics, genetic stamps, and genetic signatures. So let's see if you can reiterate that. Anyways, let's go to this clip. And just, Ian, we love you. You, Luca, speed of sound in the house. We love you all. Even though you guys are dodging, ducking, dipping, diving, dodging, all over the place. It's fine. We still like you. You guys just have to own it. Yeah, I'm just kidding. You don't have to join. But you could always reiterate the prediction in the chat. Okay, so I forgot if I put the audio on. So let's screen share. So guys, we spent the first about half an hour to 45 minutes debunking critics like Dan on mitochondrial DNA Y chromosome. And then we touched on viruses, the benefits of viruses, bacteria, retrotransposons, ERVs, ALUs, so on and so forth. So I'm going to show, see, Ian, I appreciate that. You and Sal are my favorites. I love your retort. Hey, you got to keep it fun. We got to keep it fun, you know, keep it entertaining. So we love our atheist buddies. Lena Powell has Lena, you've been such a blessing. And Lena versus Ian. Poor Ian wouldn't stand a chance. Ian's proud of his trash talking for form. Hey, we got thick skin, and our atheist buddies got thick skin too. And let's screen share here. I want you guys to see. So I'm going to go on mute again. Make sure there's no background noise. But I just want to point out though that, for example, a prediction on, say, the Khoisan peoples. Hey, I'm so confident with the mutation rate, I'm going to predict that these people groups who we don't know their mutation rate yet, I predict they're going to mutate this much. Okay, their DNA is going to change this much in however many generations. Now we actually have to test it, get their DNA. We'll see if it's true or false. That's called a future testable prediction. Now, there's been predictions on the history of civilization, which some on the Y chromosome have come true, and they're coming true more and more every day. So also prediction after prediction on mutation rates in animals as well as speciation rates. So anyways, here we go, guys.
And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless.
Well, because I've even got another paper here, and I can post them in the chat after. It says ERVs frequently act to distribute regulatory information and thus confer genes with new patterns of expression and function. And I mean, I can go on and on, but for sake of time, I'm not going to. These, even if we just look at the greater class of retrotransposons, the fact that they're extremely good for us, they can, like, that's why some of them are called jumping genes. They can jump around in the genome, and they can turn on and off various types of genes. And there we go, paper after paper. Oh no, it's just one or two, functions that were easily co-opted, even though we have no evidence for the co-option. No, the evidence suggests that these are all created units of DNA function. And just some of these comments with our good friend Speed, we love you. We hope you're doing well. But look at this. Just give me one prediction. I mean, you want a prediction in geology with the cold slabs. You want predictions in astronomy with Dr. Russell Humphreys. You want the huge number of predictions coming from Walt Brown's model. We just had Bob Enyart and Brian Nickel on, where they talked about it for 2 hours. You want the number of massive predictions coming in genetics from Dr. Jensen, Carter, Sanford. Let's see, here's one thing. One paper there on the Y chromosome. The history of civilization. One paper was just shown on screen. Okay. Let's see, because I've explained it 1,000 times. Let's see if any atheists in the chat, okay, can reiterate Dr. Jensen's history of civilization predictions on the Y chromosome and the mitochondrial DNA. Let's see if they can. Sure. So Speed says, just give me one. Just give me one. So that's the one I'm talking about right now on a history of civilization prediction on the Y chromosome mitochondrial DNA. Just came out with a paper in December on the Y chromosome, detecting signatures. Okay. Here's the thing. If these phylogenetic trees, if what we see in the Y chromosome and the mitochondrial DNA really is a reflection of hundreds of thousands of years in deep time and not roughly 6,000 years, then what we would see for the most part is a lot of genetic noise. Okay, you wouldn't be able to differentiate between noise and signatures. But guess what? Jensen made this prediction. He just came out with a beautiful paper in December showing just that. He is reading from the Y chromosome, from the mitochondrial DNA. He is showing demonstrably that this is a reflection of just thousands of years. Anyways, I can't let this get derailed for too long, answering the same questions 1,000 times. So the next clip I want to show you guys is Dr. Sarfati reiterating and adding a couple new points, solid points to what I was saying earlier on viruses, bacteria, and the benefits of them. See, and speed of sound, he doesn't want to admit that what we're seeing is genome-wide, at least 60, 70% activity in our genetics, as well as these variation-inducing genetic elements, which were never predicted to have the incredible functions that we find them having today. But yet this was all predicted by intelligent design advocates, those that believe in a creator, and those that don't assume junk. If you looked on under the hood of a car, are you going to assume this is all junk just because you don't understand it? Have a kid look under the hood of the car and say, hey, start tearing out things that you don't understand what they're there for. He'll start tearing out everything. No, we understand so little of the DNA language. So creationists have predicted a lot of DNA function. Dr. Jensen has looked at specific DNA positions. He's got specific predictions in print where, hey, this specific DNA position is going to have this function. Or if you mutate, okay, you mutate this specific DNA position, it's going to result in disease. And sometimes the specific disease can be named. Prediction after prediction on function, so-called non-coding DNA versus coding DNA. So God bless you too, Lena. God bless you too. And awesome. I'm glad Ian says he likes the new shirt. I appreciate it, my man. I appreciate it. So lots of good people in the chat. Doki Doki, thanks for the super stickers. You guys are great. So I want to show this clip real quick, and then we're going to go over a paper as well. And okay, here we go. Him say, good to see you as well. Says genetic entropy is a good, yeah. Here's the thing, genetic entropy. They got no explanation for these nearly neutral mutations that accumulate from generation to generation, just slightly deleterious. Selection can't see them. That's the whole point but behind neutral theory of evolution. They build up like a hidden reservoir for evolutionary advancement where they can be called upon later for change, adaptive purposes, for example. But no, it turns out they're slightly deleterious. They're effectively neutral, and they slowly degrade us. This has all been confirmed through thousands and thousands of numerical simulations. Can't be refuted.
Evidence against young Earth creation. What's the
okay? Most bacteria and viruses are good for us. I mean, you've got probably slightly more bacterial cells than human cells in a healthy human body.
Right.
Well, I think it's about 30 trillion human cells and about 38 trillion bacterial cells. Most of them are in your large intestine, the colon. Okay, so they're good for you. As soon as a baby's born, the large intestine is populated with trillions of these bacteria. The immune system is working really, really hard, much harder than it works when your baby's vaccinated. So the hardest thing it has is to try to control these germs inside your gut so they stay where they're meant to be. But they're doing a good thing. They're actually helping you digest some food, you see. So they're good for you. And most bacteria are good for you even now. But there are ways since the fall happened that some good things could become bad things. I mean, it seems that some of the parasitic bacteria are related to bacteria which are not parasitic, but the bacteria have lost about 80% of their genes.
Wow.
Which means they have to be parasites. They can't get the nutrients unless they're parasites. But the thing is, they've lost a lot of information. In fact, you look at parasites, they seem to have all been degraded. They're genetically deteriorated in relation to some beneficial germ. There's some machinery that gets lost sometimes, and the thing has no choice but to be a parasite. And even viruses, you've probably got 10 times more viruses than you have bacteria in your body, because the viruses are helping to regulate the bacterial population in your intestines. So they're doing a good thing as well. These are bacteriophage viruses that are regulating your bacterial population, and they're all over the earth. I mean, in the sea, you've got bacteria, viruses regulating all these things. In fact, one secular article earlier this year was saying that, if we eliminate all viruses, you'd probably be healthy for about 1.5 days before you started to die. And I think even coronavirus, I mean, that seems to be a benign bat virus that's got out of its proper habitat.
Right.
Influenza seems to be a benign avian, is it waterfowl, ducks and things like that. It seems to be a benign thing. It does some good in the duck, but when it gets out of the duck population, comes into human, it can be very nasty.
So when these viruses jump species, or as you said, jump from their original habitat, that's where they can become harmful. That's when they can burn hot and fast kind of thing.
Yeah, that's a big thing, yeah. And Dr. Carter's written articles about that. He wrote on the coronavirus, and that's the thing called zoonotic viruses that originally were in other organisms. It seems that bats use viruses to keep down cancer. It seems like they kill cancer cells quite well. They do some good for the bat, but when they get out of the bat, they do some harm for us. Ebola and the coronavirus, the COVID-19, SARS-CoV-2 does harm to us. And Ebola does harm to us, but it probably does some good things for the bats.
Right. That's a really really, really good point. To us, I'd imagine our immune system, it's foreign to us. Therefore, we can't, our body doesn't recognize it. I love the answer you're giving, because this is an argument that they sometimes pose to us as like a knockout argument, when in fact it makes me, based on what you're saying, Dr. Sarfati, it makes me wonder, imagine being in a world without bacteria and viruses. Like you said, we'd survive even a day.
And I think therefore Adam pre-fall would have had an immune system, because the immune system distinguishes between self and non-self. So if Adam had healthy bacteria in his large intestine, he still had to have the immune system to keep the bacteria where they belong. So even in the pre-fall world, there would have been a function for the immune system. Right. They wouldn't have got sick because it would have worked perfectly. Now our immune system doesn't always work perfectly because we're living in a form where Adam and Eve's immune system would have been perfect.
Awesome answer. Well, okay, guys, so I want to share an article here that's going to talk a little bit more about this, and we're going to go through it. Okay, so let's check it out here. There we go. Okay, so this is what we've been talking about. Anybody who just got here, we spent the first good portion on mitochondrial DNA phylogeny, as well as Y chromosome, debunking the critics and their arguments against independent origins. We've then gone on to this topic of viruses, viral-like elements, endogenous retroviruses, and then we're going to end it with some genetic entropy as well. Okay, so let's see. Some evolutionists allege that 8% of the human genome originated from viruses. This number is ambiguous, since different authors include different genetic elements as viruses, such as signs and lines. For example, ALU elements make up at least 11% of the human genome. Now remember, the evidence seems to suggest, guys, based on everything we've discussed, the huge numbers of papers that we've gone over, these are, and let me know how my audio is too, guys, these are variation-inducing genetic elements, okay? That's what they've originated from within. They've been created. You can have rogue agents, external escapees, internal escapees, okay? Genetic entropy can mutate a bacteria to the point where it's lost so much of its genes that it's now become bad. It's become a parasite, essentially. This is what the evidence suggests. And these arguments from this guy, Barry, his blog, he keeps commenting on my Facebook page, and I say, let's debate live. This is why he won't, though, because then you can call him out for providing evidence that doesn't help differentiate between the models as to whether or not these are created units of DNA function, whether or not they've originated from within or from outside. Okay, no. We need to explain that which can differentiate the explanations and models. If I were to come to a debate and say, hey, Luca, the sky's blue, creation is true, you'd say, so what? Evolution can also explain why the sky is blue. Or I were to say, hey, speed of sound, the earth is a sphere. You're not related to a banana plant. You're made in the image of God because the earth is a sphere. Speed of sound would say, so what? I can also explain from my model and my starting point why the earth is a sphere, why the sky is blue. We need to focus on that which can differentiate the two models. And all the arguments that we see for endogenous retroviruses and retrotransposons, okay? All these arguments are not helping to advance the debate. Our major prediction, okay, is function, benefit to these variation-inducing genetic elements. And that's exactly what we see. It's exactly what we find. It's exactly what we've predicted. And dokie dokie, I got to say, coffee would be good right about now. Since time flies by, I said, we're just going to do this for an hour, and already we're going on an hour and a half. Okay, so let's keep going. According to another study, 22.4% of the genome is covered by endogenous retroviruses. Well, I read 3 sentences, guys, and I went on a 3-minute rant, so I'm going to try and avoid that, or we'll never get through this. So let's follow along. They claim that this came about when viruses infected humans and inserted their DNA into the human genome. This process is believed to have taken place over millions of years of evolutionary time. However, This has never been scientifically proven by direct observation. And remember, guys, this co-option of function, never observed. I have now officially had about 100 debates. Most of those, the ones I should say that I've asked, hey, show me observable evidence. Show me a technical paper of a non-functional ERV going from non-functional to functional. And guess what? It's never been presented. One biologist I debated, he at least had a bachelor's in biology, I think it was, on T-Jump's channel. He said, hey, listen, if I could present, if I said I had a paper that showed empirically in the lab in real time the co-option of an incredibly important function that we find these ERVs are having and exhibiting, I'd be lying because they don't exist. Doki Doki, thank you so much for the super sticker. Thumbs up, I like it. Okay, so here we go. Researchers from the University of Massachusetts Medical School have been studying the spread of koala retrovirus as it infects the genomes of koalas from northern to southern Australia. Based on this, these researchers claim that a similar process could have also happened with other viruses in our own genome in the distant evolutionary past. How do retroviruses such as KoRV manage to insert themselves in the genome of a species? A retrovirus is a special kind of virus that inserts its own genome into the DNA of a host organism. Hence the retro, right? That's why they call them a retrovirus. A well-known example of a retrovirus is HIV. But here, this is important, guys. Watch this. This infects our immune cells, okay? But usually retroviruses spread horizontally from individual to individual, sometimes causing illness as they go along. But this process does not allow them to insert themselves permanently as part of the genetic material of a given species. Also, guys, lead to incredible benefit and function in determining cell types and gene expression, in immune responses, in epigenetics. I mean, these evolutionists are grasping at straws. What researchers want to see is whether the virus can transmit itself vertically, right? There's a difference between horizontal transmission and vertical transmission. Okay, guys, I want you to pay close attention to that. From parent to offspring. This would mean that the virus has become a permanent part of the genome of the species and will persist in the species in the long run. During horizontal transfer, viruses spread between the body cells of two individuals, but during vertical transfer, viruses jump from a somatic cell, right, a regular body cell, to the germline. The germline is made up of reproductive cells, sperm and eggs. If viral DNA is incorporated into the genome of the cells that give rise to the sperm or egg cells, then the virus will survive to spread in following generations. And remember, guys, this is just the start of the problem for them. The next major problem is the actual co-option of these incredible functions that we've detected, that we've predicted, that we've expected if they've actually originated within the genome, and if they are created units of DNA function. Then they want to ask you, well, why do we share so many retroviral elements with the chimpanzee? That's because we share a lot in terms of morphology, anatomy, physiology, and genetics with the chimpanzee. So if these are created units of DNA function, used for function in the DNA, that is then reflected by the phenotype, then of course we're going to share those similarities with other similar organisms. That's why we also have ones we don't share, because we're not exactly the same. But we're close enough that we're going to share a lot of these variation-inducing genetic elements. We'll share more with a chimpanzee than we do with a creature that's not quite similar to us in terms of the way the chimpanzee is. Okay, so let's keep going here. Those viruses, which have become totally fixed within the genome after a long time, are called endogenous retroviruses. That's where they get their name. According to evolutionary theory, in this manner, the genome would be much like a molecular fossil graveyard. In this graveyard, we would find the remains of different kinds of retroviruses as a sort of genetic fossil record of infections by retroviruses over millions of years. See, it's like when they look to the fossil record and they're looking to the transitionals, right? They're looking to fossils. Just like in the genome, the evolutionists want to look to the genome for fossils. To them, it's like a fossil record. Let's look at these genetic mistakes like the pseudogenes. That's why I always say the head-to-head prediction is DNA function. You know, if created heterozygosity is true, and God really did front load the genomes of living organisms with functional and helpful and beneficial diversity, including these variation-inducing genetic elements, then we would expect just that function, okay? So that is a direct prediction coming from the creationist model, while evolutionists believe this has all been a process of genetic mistakes, evolutionary leftovers, genomic fossils. So that's what we should find if evolution were true. And that's why it's funny that that which they point to as being the evolutionary leftovers, as being the genomic fossils, it turns out that no, they are functional DNA elements because they are beneficial to life. They are functional to life. They are crucial to life. Head-to-head prediction, and it's the creationist predictions that are being fulfilled more and more every single day. Why would the cell replicate a bunch of junk? A bunch of worthless junk. Not to mention the fact that these pseudogenes or your ALUs, your ERVs, if they really were just evolutionary leftovers, genomic fossils, a molecular fossil graveyard, they'd be mutated to oblivion. A lot of what we see is conserved, preserved, okay? So let's see. Where were we? The older the retrovirus, the more copies it has in the host genome, since it infected the genome much longer ago, or so the story goes. This is how evolutionists try to explain the presence of 30 to 40 different families of viruses present in 1 to 1,000 copies each, allegedly introduced over the past 30 million years, according to the evolutionary timescale in the human genome. So this is going to be important, guys. Once inside the human genome, several things can happen to retroviruses. Usually they can lose their function and degenerate into functionless DNA. They can also cause illnesses such as cancer. This happens when the retrovirus disrupts a gene by inserting itself right in the middle of it. This would be like inserting a long paragraph in Russian into the middle of an English novel. The result would be unreadable. Evolutionists amazingly claim that retroviruses, Brother Brandon, good to see you, man. Good to see you. Always a pleasure. Yeah, they deny reality. They deny reality. Here's the thing, Brandon. The same people, okay, that say there's no evidence for genetic entropy, it's not real, it's not reality, because they're denying reality. In 1 week, I discussed genetic entropy live with Leophilus, David Neff, and Mr. Parker, who goes by Godless Galaxy. 3 critics that say in live streams when they're with their evolutionist buddies that there's no evidence for genetic entropy. Then you get them in live discussion, live debate, which is why this Barry guy only wants to be a text warrior instead of have a live discussion, because guess what? You get them in live discussion and suddenly they don't have any answers. They crack. And suddenly they have to go from being so condescending and overconfident to, well, I'm not a geneticist, and that's fine. But guess what? Population geneticists recognize there's a problem of mutation accumulation, which is why they're coming up with artificially contrived rescue mechanisms like mutation count mechanism, synergistic epistasis. Why are they coming up with mechanisms to explain something that doesn't exist? Answer me that one, Leophilus. That's too funny. Anyways, let's keep going. This would be like inserting a long paragraph in Russian into the middle of an English novel. The result would be unreadable. Evolutionists amazingly claim that retroviruses can also be, so here's this, can be reprogrammed, right? With no evidence at all. Evolutionists amazingly claim that retroviruses can be reprogrammed or repurposed to take part in cellular processes. One frequently cited example is the syncytin gene, which this Barry guy used as an argument in his review of my book, which allegedly originated from a retrovirus and which was recoded to take part in the fusion of certain cells to form part of the placenta. This is all retrofitted. It's all storytelling. It all assumes their story as compared to the important question as to whether or not these are created units of DNA function. Okay. Let's see here. I want to go down to, let's see here. Okay. Does viral invasion of host genomes make sense? So to read the whole thing, guys, I'll post this in the description, but it's kind of long. So for sake of time, I'll skip down to this part. The evolutionary picture of viruses invading the genomes of animal species and slowly integrating as functional units into their host genome over millions of years runs into numerous problems. First, the spread of KoRV within koalas has been happening for only 100 years, not millions of years. This time frame better fits the biblical timeline rather than the evolutionary time scale. Remember, a lot of these that have gone bad, they have gone rogue, they are internal scapees, okay? Mutation can do this. Mutation can make a beneficial bacteria, it can turn it into something bad, something harmful, a parasite essentially. Second, if KoRV is useful in the long run, then why does it cause such debilitating diseases such as cancer? The researchers studying the KoRV have even suggested producing a vaccine against the retrovirus to save the koalas from extinction. How is it that so many inactive copies of KoRV survive in the koala? Jungle jargon. Yeah, it's that they talk such a big game, scoff at Dr. John Sanford, right? But then when you get them into a live discussion and you point out the fact that, hey, listen, you can't invoke selection to remove that which is unselectable. So what type of mechanism is going to remove that which is unselectable but still slightly deleterious, okay, to the genome over time? And they've got no answer because there is no answer. Genetic entropy puts shelf lives on genomes. Let's keep going. Around 8% or more of the human genome is made of retroviruses. If the KoRV is retained in the koala genome as junk DNA, it would most likely have been removed from the genome. Remember I was talking about this earlier? It's costly for the cell, guys, to replicate so much junk. The cell would rather get rid of it, or natural selection would get rid of it. If it's just a waste, it would cost the cell too much energy and material to keep the junk DNA intact for an indefinite length of time. This would be like printing a book with extra pages of gibberish at the end over and over and over again. Isn't that right, guys? It would be superfluous to keep junk DNA in the genome for such a long period of time as deep time evolutionists would say we've been around for. Yeah, just keep all this junk DNA, all this worthless, useless evolutionary leftovers in there. It'll be wasteful for cellular processes without eliminating it. It is much more likely that ERVs such as KoRV already had some function in the genome to begin with. Remember, if they've originated from within, they're created. You can get internal escapees, external escapees, good variation-inducing genetic element to bad variation-inducing genetic element for a number of reasons that we've touched on. Right? Here's the GAG gene, POL gene, ENV, LTR. You know, Luca was talking about how can we identify these? Yeah, that's not the debate. That's not the debate. We agree on what they are, but we don't agree on what they're for. We don't agree on the origin of them. And these are the only arguments you're going to get from the evolutionist side. They're not going to make predictions. They've never made predictions on the function. Guess what? We'll put it on print. Hey, this DNA position is functional for this reason and this purpose. Hey, this specific type of creature, because of its function as being, say, a mammal, or its function in physiology, it's going to have this function versus this animal type, which we say is not related. It's going to have this type of function in a similar genetic sequence. We can make these types of predictions. We can say, hey, this genetic sequence or this DNA position, if you mutate that in a mutagenesis experiment, it's going to result in a disease because it's functional. It's there for benefit. It's there to be helpful to the organism. It was created. Okay, we can make those predictions, and we've got a ton. You got people like Speed of Sound who say, show me one, I'll show you 1,000. Okay, that they can't counter either. That's the structure here of this KoRV genome, right? You got the GAG and PAL genes are responsible for inducing genetic variation. The ENV gene codes for the outer envelope of the KoRV virus, but can be associated with genes in the host genome. This Barry guy with his blog, his ERV blog, and his comment about the Phoenix experiment, which I decimated, he won't be able to respond to this. He can come debate me on this if he wants to. The challenge is there. Also, it is a wide stretch of the imagination to say, this is what I was implying earlier, guys, that it's likely to disrupt the existing genes, okay, as compared to improve it or make it as functional and important to the genome as we know these elements are, okay? Because if you had a painting and you had a painter and you put a blindfold on him and you said, finish this painting, make it as best as you can be, okay? And he started just applying just random brush strokes, doesn't even know what he's doing, can't see anything. What's going to happen is he's going to ruin the painting. He's going to mess it up. It's not going to make it better, okay? Lastly, why is it that host piRNA already exists in the genome with a sequence? This is important. With a sequence. Thank you, Doki Doki. I appreciate it. Hope you guys are enjoying this. Hey, 5:30 in the morning. We got 20 people here. This is awesome. That's why I love my audience and supporters. You guys are a bunch of night owls and party animals like myself. All right. Already exists in the genome with a sequence, which is complementary to that of the GAG and POL genes. Interesting. It is as if the host genome had been able to look ahead and had already come up with the piRNA sequence in order to counteract and deactivate the GAG and POL genes of the invading retrovirus. But evolution, right? This is where they want to give evolutionary mechanisms a mind, because they don't want to attribute what we see in our genomes to a forward thinker, I'm sorry. No, they need to apply evolutionary mechanisms. They need to apply a mind to them. But evolution is built upon blind chance and cannot know ahead of time what kind of challenges will face the organism. This is like stumbling upon a tribe of isolated forest dwellers, one of whom can speak perfect English with you. See if Speed can answer that one in the chat. It is more likely that piRNA already existed within the host genome as a design element to keep the levels of the KoRV in check. All right, here, guys. Here's why creation is the better explanation for the 100th time. And we're going through technical papers from the secular side, technical papers and articles from the creation side. We're going through clips. We got a ton here showing that evolution fails on all levels, guys. It is possible to reinterpret retroviruses according to our model. Accordingly, so here we go, guys. This is what I was saying earlier. Accordingly, and I said the same thing to Team Skeptic in our debate. And his response was, well, there's no evidence for that. After providing all of this evidence that I've done tonight, showing that retroviruses could have originated from within the host genome instead of invading it from the outside. This had to, guys, listen. This had to have been the case in any scenario, since viruses are dependent upon host cells. Remember that. This had to have been the case in any scenario, since viruses are dependent upon host cells. Any evolutionists in the chat? Give us any reason to believe that they have come from the outside, when in fact, viruses are dependent upon host cells. This means that the cell's genome, guys, I can't emphasize this part enough. This means that the cell's genome had to predate viruses. Had to. God created the first people, the first organisms, and front-loaded them with these variation-inducing genetic elements. After the fall, many have gone rogue, many have gone bad, many have escaped internally, externally, but no, they've all originated for good. And that's what we see. That's what the evidence suggests. So now the similarities and the hierarchies that they form make sense according to our model, since of course we're going to share a lot of these variation-inducing genetic elements with the chimpanzees and other organisms that we share a lot with in terms of anatomy, morphology, physiology. This is just common sense. It's what's predicted. Viruses are incapable of surviving without a host cell. That is, the ERVs are more likely rogue products. Remember this. The ERVs are likely rogue products of the genomes of their hosts. And the parts of the genomes that look like ERVs are then seen as designed to do what they do, and they do it well, guys. Research will increasingly reveal functions. Isn't this funny? Here's a prediction right here that we are okay and confident enough to put on paper. Research will increasingly reveal the functions of these misnamed retrovirus elements. Dr. Ian Macready, at the time researching the retrovirus HIV, suggested this way back in 1999. Since viruses take up such a large portion of the genome, they must have some sort of function. Viruses are capable of transporting genes besides their own between bacteria, for example. Isn't this amazing? According to the variation-inducing genetic element, or VIGE hypothesis, coming from our model, certain genes in host genomes do not originate from retroviruses from outside. Rather, these genes are influenced by transposable genetic elements, which can alter the regulation of genes. When these transposable elements leave copies of themselves behind, they ensure that the new variation they brought about can be inherited. The transposable elements which cause the variation within the host genome are the gag and pull genes. The gene that they regulate is the ENV protein. The syncytin gene is attached to the ENV gene of the so-called ERV-W element, which is expressed in a part of the placenta called the syncytiotrophoblasts. Say that 10 times fast. Yet another regulatory transposable element called the trophoblast-specific enhancer is responsible for expressing the syncytin gene only in the placenta and nowhere else. Syncytin is essential for successful pregnancy. So how did mammals reproduce before the putative ERV-W infected the mammal line and then fortuitously developed this function? It is all rather fanciful, wishful thinking on behalf of the evolutionists. Isn't that funny? So, guys, I want you to finish reading this, but read the whole thing for yourselves. This is a fatal blow to the evolutionists who want to keep claiming that viruses, viral-like elements, retrotransposons, ALUs is evidence for evolutionary theory. No, this is not evidence for relationship between banana plants and whales. No. Speed, Luca, you're not related to a strawberry. I understand when you pick up a strawberry and you look at it and you say, look at all this similarity. You know, when I get angry or upset or embarrassed, sometimes I turn a little red like that strawberry, but that's not evidence that I'm related to the strawberry. Guys, you're made in the image of God, and that's what this evidence suggests. Variation-inducing genetic elements is what we are looking at. Okay? From within, certain mobile retrovirus particles originally could have moved from site to site within the genome, thereby changing the way genes are regulated within the host genome. It makes much more sense if retroviruses were originally designed as part of the host genome and escaped from the cell after the accumulation of mutations. All genetic entropy, genetic degeneration. Let's see here. Evolutionists claim that retroviruses such as the koala virus are invading. Luca, I do too. And they're good for you. invading the genome of their host species. They say that this way these viruses can help the evolution of the host genome by inserting novel DNA into it. This idea is rather implausible. It goes against the logic of natural selection because the koalas become sick due to viral infection. Therefore, these koalas infected with the virus would be eliminated together with any copies of the viruses within them. But there is a second hurdle at the cellular level. The koala virus must jump before it can fully get integrated into the genome of its host. This is a molecular selection system, which eliminates genes which the cell does not recognize as its own. This involves special RNA molecules which bind to the virus genes, therefore removing them from cellular traffic and making it impossible for them to become a permanent part of the host genome. All right. That was a long one, guys, but an important one. We like to leave no stone unturned. So let's see how the chat's